Hello, everyone, and welcome to episode 870 of the People Behind the Science podcast. I'm your host, Dr. Marie McNeely, and today we are excited to be resharing our interview with our guest, Dr. Sharlene Santana. Listeners, Sharlene is an integrative and evolutionary biologist who explores questions about evolution from various perspectives. She's working to understand why some groups of organisms are more diverse in terms of their number or their species or appearance or behavior. And in particular, she's interested in bats. So there are over 1,400 species of bats that fulfill a variety of ecological roles, and much of her research focuses on diversity in bats. And in our conversation, Sharlene shared some excellent stories about her life in science. So I hope you're ready to meet another one of our amazing people behind the science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to People Behind the Science. Today, I am delighted to be speaking with our guest scientist, Dr. Sharlene Santana. So, Sharlene, welcome to our show today. How are you?
I'm doing well. Thank you so much for this invitation, and thank you to your listeners for tuning in.
Absolutely. Well, we are excited to hear more from you about your life, about your science. But before we jump into the interview questions, let me first start by giving our listeners a little bit about who you are and how you got to your current position. Listeners, Sharlene is Associate Professor in the Department of Biology at the University of Washington and Curator of Mammals at the University of Washington's Burke Museum of Natural History and Culture. She completed her undergraduate training in biology at the University of the Andes in Venezuela, and she was awarded her Ph.D. in Organismic and Evolutionary Biology from the University of Massachusetts Amherst. Afterwards, Sharlene conducted postdoctoral research at the Institute for Society and Genetics at the University of California, Los Angeles, and she joined the faculty there at the University of Washington in 2012. And in our interview today, Sharlene, we are excited to get to know you both as a scientist, but also more as a person. So can you start by telling me and our listeners what you like to do when you're not doing science?
So I should say both my husband and I are scientists. So we live in a very science-heavy household, but we like to spend most of our free time together trying new restaurants around here in Seattle. And we have a big dog, so we like to take him hiking and swimming. I also enjoy good storytelling. So that can be either reading a book or watching a movie, watching a TV series or a documentary, or even enjoying a dance performance. I'm a really big fan of ballet. So whenever I have the time, I like to go to a ballet performance. And I also like listening to music and podcasts. But I would say that my favorite thing to do when I have enough time is to travel to other countries and learn about their cultures.
Oh, that's amazing. It sounds like you've got quite a bit to keep you occupied outside of your science. But I know many of us, myself included, have been maybe watching a little bit more TV, movies, etc., than we usually do. Is there something in particular that you're hooked on at the moment?
Definitely. Yeah, we are watching some Netflix shows. We just finished watching When They See Us, which was a very heartbreaking, but also a very eye-opening show that we enjoy watching.
Excellent. Well, listeners, we'll have to put this on our watch list. I love it. And we touched on some of the things that you do outside of science, but I do want to talk about your research as well. You've got some fascinating projects going on. So how do you describe your work to people who aren't familiar with science?
I am an integrative and evolutionary biologist, which means that I explore questions about evolution, but from a variety of perspectives. But mainly my research tries to understand a very fundamental question in biology, which is why are some groups of organisms more diverse, both in terms of their number of species or the way they look like or behave? So to answer this question, I focus on mammals and particularly bats, because bats have radiated into over 1,400 species and they perform a wide variety of ecological roles in their ecosystem. So they have also evolved these roles multiple times throughout the course of their history. And that makes them a really good, say, natural experiment to try to answer questions about evolution. So to understand why bats or even some groups within bats are highly diverse, our general approach is to first look at how specific aspects of the, say, anatomy, behavior, and ecology of bats are different across species. And that allows us to identify which groups have greater diversity. And then with that information, then we come up with hypotheses which try to explain why some of those groups might be more diverse than others. And then we use a wide variety of tools. That's where the integrative biology comes into place to test those hypotheses. I'll give you an example so this doesn't sound too esoteric. So say we find that a group of bats has a greater number of species, and we looked at the different anatomies of these bats, and we find that they have a specific type of skull shape, and at the same time, they have a specific diet. So what we do then, that gives an idea, okay, maybe there is a relationship between the skull shape, the diet, and the number of species. And we test that by, for example, building computer models of their skulls that simulate biting. And that allows us to tell if those skull shapes are better at eating certain foods than the skulls of the bats that are not as diverse. So we use all these different approaches to understand mechanistically how different groups of bats might have become more diverse.
Certainly. And I think me and maybe our listeners out there as well sort of have this almost archetype in our minds of what a bat looks like, but it's way more diverse than I imagined. So can you talk a little bit about kind of the range of bats that you're looking at in terms of size or features?
Absolutely. So like I mentioned, there are over 1,400 species of bats. And I think most people think of maybe a handful of bats. Mm-hmm. And within that 1,400 species, you have animals that are as small as a cotton ball and weigh almost about the same, all the way to bats that have a wingspan of up to 6 feet, and everything else in between. And all parts of the body are also very diverse. You have species that have super long snouts. For example, the bats that feed on nectar, they are kind of like the hummingbirds of the bat world, all the way to bats that have very short faces, even shorter than, say, the face of a monkey. And those are bats that are feeding on hard fruits. They have a lot of diversity, both in terms of size, anatomy, not just of the skull, but other parts of the body. And that's reflecting the different ecological roles that they have in nature, from eating fruit and nectar, but also insects, other animals, fish, anything you can think of.
Well, Sharlene, I look forward to chatting more about your research as we go through our conversation today, but I'd love to take a moment here at the beginning to talk about motivation. I think all of us can use a little extra motivation and inspiration these days. So when you think about your own life, is there a particular quote or a saying or just maybe a force that is particularly motivating for you?
Definitely. So my favorite motivational quote is actually just 2 words: everything changes, which I think derives from the quote, "Nothing endures but change," by the Greek philosopher Heraclitus. And it's also related to this Buddhist concept of impermanence. So I found these 2 words, everything changes, particularly comforting during difficult times, but also it's a good reminder of not becoming too attached to things or ideas or places. And as a biologist, it's also a good reminder of the ever-changing nature of the natural world and the environment. Kind of a useful lesson, both for my personal life, but also my professional life as well.
Oh, I love it. Well, wonderful quote there. And I'd love to talk next about some of the people, Sharlene, who might have motivated and inspired you over the years. So are there particular people who rise to the top of your mind who served as role models or mentors or even just historical figures that you've looked up to?
There are many different people, I think, that have been inspirational for my career, both in terms of the way they approach their science or their personality. But I wanted to highlight one of those people, specifically Dr. Elizabeth Calco. She was a professor at the University of Ulm in Germany. And Ellie, as we used to call her, she was the first international female scientist that I ever met back when I was an undergrad in Venezuela. And she was super kind and helpful as I was developing my research project there, and very patient with me. I was pretty starstruck to meet the scientists whose papers I had read and enjoyed. But her career was also just so inspiring. She was a very outstanding scientist and experimentalist. But at the same time, one thing that I really liked about her work is that she really thought about and incorporated information about the natural history of species. She was a person who spent a lot of time in the field looking at what animals are actually doing. So she informed a lot of the work, the hard experimental work based on that information. And again, she was also just this very kind and enthusiastic person who was very generous with her time and her ideas. And she was a good mentor also to me. not just back then when I met her the first time in Venezuela, but over the years as we met again in different instances, or even when we collaborated later on.
Oh, that's wonderful. And I know a lot of biologists have their interest in science sparked by this sort of being drawn to animals in the natural world. Was this the case for you?
Yes, to some extent. I was always interested in animals when I was a child. We had a big backyard and I lived in the tropics. So that meant that we had a lot of animals that visited us, a lot of tropical birds. And we always had pets in the house, had a lot of dogs. So I was always very fond of animals and the natural world in general.
Very cool. So then how did you start to maybe develop this interest in science and start to think about it as a career option?
I had to decide this pretty early on. As you mentioned, I did my undergrad at the University of the Andes in Venezuela. And over there, the undergrad system is very different from here in the United States, because when you go on the biology track to be a scientist, that's separate from, say, the biology track for people who are going to go to med school or vet school. So you had to decide pretty early on that that's what you want to do. But I think I was pretty sure that that's the career path that I wanted to take. But when I started my undergrad, a specific area of biology wasn't too clear to me. I thought I wanted to be a molecular biologist, but that changed pretty quickly after I had some really great experiences with classes that exposed me to field work and anatomy and behavior and evolution. So that really kind of shaped my interest into the specific areas of biology that I wanted to study. And again, over there, for your undergrad, it's mandatory that you have to do a research thesis at the end of your degree. So I had that opportunity to further explore those interests. And I worked with a professor who worked on bats, and that's where I got hooked on bats by going to the field with him. And I did my project on the feeding behavior of a fruit bat, who was at an interesting evolutionary position in the tree of life of bats. So we had some evolutionary implications of what I would learn from that research. And doing that project was really great. It showed me how fun it is to do science, but also how challenging it might be. And while I was doing that, I was reading a lot of papers from all these different researchers. And that's how I came to know the work of who became my Ph.D. advisor, Dr. Betsy Dumont, when she was at UMass Amherst. So I applied for her lab and got in and did my PhD there, looking at the cranial morphology, feeding behavior, and diet of the neotropical leaf-nosed bats, which is a really cool and diverse family of bats. And that also, again, gave me a lot of tools and generated many more questions, some of which I still continue to explore today. And when I finished my PhD, then my next goal was kind of build up my toolkit for the rest of my career, but also I wanted to kind of get out a little bit of my comfort zone since I had been working on bats for so long. And I went to the Institute for Society and Genetics at UCLA, where I got a fellowship to work on the evolution of facial coloration in primates. So that project integrated some behavioral aspects and also some anatomy aspects, in this case, coloration. But also, while I was there working with Michael Alfaro, who was my mentor, I also learned a lot about phylogenetic comparative methods, which are just statistical tools that allow us to answer questions in evolutionary biology. So that gave me a little bit more training and more ideas to incorporate into my research. So I was there for 2 years until I got my position at the University of Washington in 2012.
Very cool. And I know your position is unique and actually really cool because you're split between faculty and research and also this position as a curator at the museum. So can you talk about this balance?
Yeah, it's quite interesting because this was the kind of position that I dreamed of having when I was in grad school. But my professors would tell me, oh, those positions are very rare. Don't set your heart to it. And then it worked here at the University of Washington. And it presents a really great balance for me because, as you mentioned, it has a very strong research component, which is what I love doing the most. But also the curatorial component of it gives us this wonderful access to specimens, but also some ability to shape how these collections are growing. So a lot of the research that we do in my lab now involves not just studying bones, but also we study muscles and other soft tissues. So having the ability to have a say in how the collections are growing helps us build those collections in those directions with different kinds of specimens that are useful, not just for my research, but also are going to be useful for morphology and evolutionary studies in the future. And the other aspect of the curatorial role is outreach and education, public education, which is something that I really enjoy, especially as somebody who works on bats, who are very misunderstood creatures. This gives me a great opportunity to do a lot of PR for bats and how important they are.
Certainly. So was it difficult transitioning into this curator role? Because I think that's maybe not something that your postdoc or your Ph.D. has necessarily prepared you for in many cases.
Yeah, that is true. I didn't have a specific curatorial experience. I had worked in my PhD with specimens, and I have done prepping of specimens, and I was familiar with working with museum specimens, but the overseeing role is something that is completely new. But at the same time, I think just being a principal investigator for a lab, everything is new anyway. So there's just so many things that we are not trained on, either to run a lab or to be a curator in this case. So I think everything for me was merged into this big thing of just being a new faculty.
Definitely. And I think these transition periods are difficult for a lot of people, but there are challenges throughout the course of your career as a scientist, and the big giant failures, but also just the small everyday difficulties. So do you have an example, Sharlene, of one of the challenges or failures or struggles that you've encountered? And can you walk us through what was going on and how you worked your way through it?
Most definitely. We've had many instances or days where we've had difficulties or, like you said, major failures. The one that comes to mind is not even just one day or one instance, but it was like a whole month where we had things go wrong. And this was when we were in the field about 2 years ago in Costa Rica. So while we were there, Costa Rica experienced, or at least that part of the country experienced, the wettest July in 60 years.
Oh my goodness.
So there was constant downpour every day and night for a whole month while we were there. And the crazy thing is that we had gone to the field already with these pretty ambitious plans. We were going to do some collecting of plants during the day. At night, we were going to collect bats to do experiments. But of course, when it's raining that badly, you can't collect bats because the bats are not flying and foraging when there's such a heavy downpour. Mm-hmm. And it was really also very dangerous for us to go out during the day just for plant collecting because the forest trails were flooded and there were tree branches falling. So it was pretty demoralizing that whole time, but we did get through it and we did got somehow data collection done. And I think the way we achieved that was just first through just sheer love for science and being adaptable. Whenever we had a break in the rain, that's when we would go out to collect in the field. We also just reshaped our plans to see where the kinds of data that were more feasible to collect, dropped a lot of plans and ideas. And then what I remember also vividly from that time is that because we had to spend a lot of time inside our cabin, I actually took to writing this fellowship to do more work on that system.
Oh, wow.
And I actually got it. So it was actually kind of productive at the end. So I was able to return last year to continue some of that work. But yeah, it can be really tough, but I think you just have to keep your eye on the prize and not lose hope.
Definitely. And I think it's so important, this idea that you brought up of being flexible and being able to sort of be optimistic and try to make the best of a difficult situation, because you didn't just sit there in the cabin moping. You were trying to find productive ways to use your time, which I think is so important. Yeah,
definitely not. I mean, sheer optimism. And I have to acknowledge that my postdoc at the time at Caliceska and my grad student, Liza Miller, who were there with me, their positive attitude and this like sheer optimism helped so much, just having people around you that also have a good attitude. And when these things can go wrong was also really helpful. I'm really thankful to them, actually. The fact that we got through it and we actually got work done and we didn't want to kill each other by the end of the field season.
Definitely. Well, it sounds like this was perhaps ultimately a success. You were able to salvage that field season and get some useful data. And I love talking about the successes. So is there a particular victory that was your absolute favorite, or maybe just a small victory that was particularly meaningful lately?
Recently, we just got a grant funded, which is a big deal. That's how we get our work done. So we got a grant funded by the National Science Foundation. And this is a very exciting project for me. It's in collaboration with the Sears Lab at UCLA. And it's going to take my lab in a new direction. So far, most of the work that we do looks mostly at external factors and how they shape the evolution, either the anatomy, behavior, or number of species of bats. But this project is looking from the inside out, and it's going to be looking at the genetics that control the development of bat teeth and how that affects the diversity of dental shapes in bats, which we can ultimately also relate to their diet. So it's a really exciting project, and we tried a few times with the proposal for this grant. So we were so thrilled that we finally got it funded, and we're going to be able to do all this exciting work.
That is phenomenal. So do you remember that moment that you got the notification of funding?
I did. Yeah, it was just a few weeks ago, and it was really great. It was a little strange too, because we're in the middle of the pandemic, and there was also a lot of social unrest for very justified reasons. So it was at the same time a moment where I was feeling very happy, but at the same time I was feeling a bit heartbroken by everything that was going on. So it felt a little weird celebrating. Mm-hmm. So it's a happy moment, but everything is strange right now.
Definitely. And I think any sort of good news at this point is so welcome. So congratulations on this grant that was recently funded. And you touched a little bit on this exciting new project that's about to be launched. Is there a project that you're currently working on that you are super excited about and want to tell us more about today?
Absolutely. So most of the work that we do in my lab is on bat evolution. But one project that I'm very excited about is on how bats themselves can act as force for evolutionary change. So this is actually the project that we were working on in that field season in Costa Rica that I was telling you about. So it's a project in which we are collaborating with two great labs, the Riffle Lab here at UW and the Davalos Lab at SUNY Stony Brook. And we're focusing on looking at the coevolution between short-tailed fruit bats and pepper plants in Costa Rica. So these bats and these plants have this very important and special relationship with each other. The bats eat the fruits of the pepper plants and they disperse the seeds, so they help these plants reproduce. And in turn, the plants are providing the bats with a constant food source throughout the year. So because of this potentially mutually beneficial relationship, we would expect that there might be some coevolution between features of the bats and features of the plants that allow them to maintain this relationship. So in, let's say, this plant-bat coevolution world, or even in general, plant-animal coevolution world, there's this idea that, for example, fruit have evolved certain characteristics to attract frugivores that disperse their seeds. So if you think of any fruit that you like, they tend to have a distinct color or a particular scent. And again, that is thought to have evolved to attract frugivores. And at the same time, the frugivores are thought to have evolved senses that allow them to perceive those fruit characteristics. So that is the framework that we're applying to this project. We're asking the question, have these short-tailed fruit bats evolved certain sensory characteristics that allow them to detect specific traits of the fruit in the pepper plants? And from previous research, we knew that the bats actually find the fruits through olfaction, through scent. So we're going to try to link the olfactory system of the bats with the chemicals that make up the scent of the fruit. So we've been collecting chemical samples from pepper plant fruit in Costa Rica. We're also collecting guano samples and tissue samples from the bats to understand their diet, so the different pepper plant species that they feed on. And then the tissue samples are to do genetic studies of their olfactory system. Actually, one fun part of this project is that we've been studying what the bats actually do and what they respond to in terms of the chemicals that make up the scent of the pepper fruit. So we're doing experiments with wild bats to see what chemicals they prefer. In this project, we are now in the point where we collected a lot of data and we're analyzing a lot of it and writing up papers. Results look quite interesting. It does seem that the bats have evolved some unique features of their olfactory genes, and that might allow them to perceive certain chemicals that are found in the scent of the ripe fruit. Because we do see that they are exhibiting certain preferences for certain chemicals that are in greater abundance in these fruits. So we have some answers, but also some questions for future work. But we do have some indication that there might be some either coevolution or specialization between these bats and these plants.
That's really interesting. And you touched on the fact that this research is being done kind of in Costa Rica and some of your other projects as well. What is it like being out there doing field research in Costa Rica?
Oh, it's the best. I grew up in the tropics, so it does feel a little bit like home in terms of the climate. What I love the most is the biodiversity. It never ceases to surprise you how biodiverse these ecosystems are, at least to me. I can go on a hike every day while I'm there for weeks and still every day just see something new that I hadn't seen before, learn something new. So it's really great. Also, the people in the places where I work are just fantastic. Costa Ricans are just very nice people, very helpful. They feel like family. So it's a really great place to work for me.
Oh, that's fantastic. So what does it look like then? Maybe an average, if you can call it average, day in the field?
Well, it depends, right? Of course, of what project we're working on. For this project that I was describing with the pepper plants and the short-tailed fruit bats, we would have some time during the day where we would go to collect plants for our chemical extraction. So this would be a hike through a trail in the forest. We would try to hit as many different habitats as we could because we're sampling different species of pepper plants. So we would do that during the day. One person in the team would then go to do the chemical extraction. The rest of the people might be processing samples or entering data. And then during the night, if that night we were doing experiments with the bats, before sunset, we would go set out our mist nets, which are the nets that we use to capture bats in the forest. We would capture some bats and bring them back to the field station to do our experiments, which we do in a tent. And we would release the bat. And after that, clean up, maybe taking some notes after we're done with that, and then go to bed for a few hours and repeat it the next day.
It sounds exhausting, but wonderful.
It is very exhausting. Usually when I start a field season, the first day, I'm just overwhelmed with like, how am I going to do this? But then you get into the rhythm and it's just so fun, at least for me, that you put all that exhaustion aside and it's really enjoyable. Then, of course, when you come back home and then you realize you have to recover from all that work for a couple more weeks.
Definitely. Well, Sharlene, it's been so cool to get kind of a behind the scenes look at your research. And I'd love to jump outside of science. You mentioned in the beginning of our conversation that you are a fan of reading and good storytelling. And I'm always encouraging our listeners to get out there, try and read for fun if you can. So do you have a favorite book that you've read that you can recommend for all of us today?
I have many favorite books. Recommending just one sometimes feels like I'm betraying the others. But thinking about your audience, I think your listeners will really enjoy Lab Girl by Hope Jahren, a book that I read not too long ago, actually. And I just found it really interesting to learn about the career trajectory of another female scientist. And even though her trajectory is very different from mine, there were a lot of instances where I could relate to what she was going through. And another of my favorite parts of the book is all the chapters that she wrote so beautifully about plant biology. It almost made me just want to go back and become a botanist. It's this beautifully written book, so I would recommend that one.
Well, excellent recommendation. Listeners, we'll put it up on our website for you to find. And we've talked about some of the great opportunities, Sharlene, that you've had to travel to different places for fieldwork. And I think travel is a big part of life in science, whether you're doing field research or just going to conferences, meeting with collaborators and colleagues. So do you have a favorite place that your science has taken you over the years?
As you can probably gather, Costa Rica is one of my favorite places. But I think one place that really sticks in my mind was Panama, which is right next to Costa Rica, so not too far. But the first time I went to Panama was when I was in grad school, and I spent several months there. So it was a pretty formative experience for me as an independent researcher. And I worked at the Smithsonian Field Station in Bocas del Toro, which is this tiny island off of the Caribbean coast in Panama. And what was so memorable about that trip is that there was just so much biodiversity just packed in such a tiny place, both on land and in the water. So every day it was just like, again, this adventure of just capturing species that I hadn't seen before, or seeing new beetles or crickets that I had never seen before. And at the same time, there were these like beautiful beaches on a walking distance that I could go to and snorkel. So it was a really beautiful paradise to work, but also a very formative experience for me. So I think that's one of my favorite places. I haven't been able to go back since then, but it will stick in my mind as one of the most wonderful places where I've been doing field work.
That sounds incredible. So was it intimidating for you as a graduate student, just kind of going out there in the field, sort of being in charge of your own project there for the first time?
It was. I had already had experiences doing that when I was an undergrad in Venezuela. So when I was doing my thesis there, but of course I was in my home country and I had my friends who would go with me to the field to help me. But definitely having this experience where it was another country, I did speak the language, which made things a lot easier, but it was a new place with a different wildlife, different culture. And yeah, where I was in charge of all the logistics. It was challenging for sure.
Definitely. And I know I am definitely more of a lab scientist, not a field scientist. I'm not necessarily the best at adapting to unexpected things happening. So I know this happens all the time in the field. You mentioned the one example with the heavy rains from 2 years ago. Have there been other instances where things really went a different direction than you were expecting, or you had something surprising happen out in the field?
Working with bats is really unpredictable. And this might be the case with lots of other types of wildlife, because you never really know what species you're going to capture on a given night. You do your best to make an informed decision about where you place your nets and how long you keep them open, etc. Right? And you are able to expect some specific species in a particular habitat, but animals do what they do every night. They might not be in that forest patch that particular night, etc. So every night is, I always describe it as it's like Christmas morning, because you don't know what you're going to get. You're checking your nets, and that night you might get 10 bats, you might get 20 bats, you might get no bats. So you might have some nights where you do everything the same that you did the night before, and you caught a lot of bats, and the next night you catch nothing. So it happens a lot for us in the field.
Oh, well, thank you for sharing some of your experiences, Sharlene. It sounds like you've had remarkable opportunities to go out and to do field research as well as research there at the museum. And I know as a scientist, you also get to work with some really great people, and you touched on some of the wonderful collaborators you've worked with over the years. And I think the people in science are often portrayed differently in the media than in how we are in real life. So we love trying to break the stereotypes that people have of scientists. So we're not always these very serious, critical people wearing white coats working in the laboratory 24/7. So do you have an example of maybe a fun tradition you've experienced, or just a funny memory that you shared with colleagues that goes contrary to some of these stereotypes that people may have of scientists?
I think it's really funny that this stereotype even exists, because I know very few scientists who are actually serious and reserved. Most of them are just very funny and quirky people. So in terms of lab traditions, the one that comes to mind is the lab where I did my Ph.D., Betsy DeMont's lab. And every year she would have a Super Bowl party at her house. And the funny thing about it is that football, I think, was the least important thing for that party. I think very few people actually watched the game. It was just a way to build community, meet with people from the department outside of it, meet their families, talk about other things rather than science. So that was really fun, especially for me coming from a different country, to see this tradition here in the States. And then the other thing I remember also from Betsy's lab is that at some point, us grad students and the postdocs, we started calling ourselves the Monsters after her last name.
I love it.
Yeah, and I'm not sure how that started, but since we studied bats, we thought it was very fitting.
Oh, that is amazing. So have you developed any traditions or things that you do with the group in your own laboratory?
It changes a lot depending on the group. We do try to have at least one barbecue every year where we have everybody and their families over. We do a lot of things with the museum too, depending on the different events that are going on at the museum. So after we're exhausted to doing a big event, we all get together and relax and have some drinks. We've done a lab retreat out here in the San Juan Islands off of the coast of Washington, which was also really fun. So yeah, we're still building on traditions. I just had a big turnover in my lab where all my grad students graduated and my postdocs finished their positions and left for jobs. So I'm going to have a new set of people now to build new traditions with.
The perfect opportunity to experiment, right?
Exactly.
I love it. Well, you are answering some phenomenal questions with the work that you do in lab, Sharlene. And we talked about some of the challenges you run into. And I think in science, there are often barriers, things like restrictions in funding, staff, technology, feasibility, time. We could go on. But if we took all of those barriers, all of those challenges and restrictions away, what is the one question that you would want to answer most?
The question I think that bothers me the most, I would say, every time I do field work and I'm walking through these tropical rainforests is how are all these organisms interconnected and able to coexist in such a diverse community? So this is a question that would definitely need unlimited resources because first we would need to know the species that live there, and many of those species have not been described. So it would be a massive undertaking in taxonomy and systematics. And then it would require thousands of studies about the ecology and the physiology and natural history of the species to look at their relationships. But if that was possible within, I guess, a single lifetime, it would allow us to answer many fundamental questions, both in ecology and evolutionary biology. And one of the most important things is that then it would open the opportunity for really effective conservation strategies of this highly diverse ecosystems that we're losing at such a fast rate.
Definitely. So given unlimited resources, is there one particular place you'd want to start this very comprehensive project in?
Well, I guess why not go big and say the Amazon rainforest?
Very cool. Well, Sharlene, thank you so much for dreaming big with us on the show. And you've shared so much in terms of your stories, your experiences, and your expertise. I'd love to end with advice. So for our listeners out there who might be going through their own scientific journeys, what is the best piece of advice that you ever received in your career that you can share with them?
It would have to go back to this experience where we had a lot of failures in the field. Perseverance and adaptability, I think, are two things that are really important in science, especially as our world is changing so much right now. I think there's a lot of instances where failure is very common, both in terms of not just experiments in the lab, but also trying to secure funding sources for our work. So being perseverant is really important in that sense. And then the adaptability is also just quite important for very similar reasons as well.
Well, wonderful message for our listeners. And we covered a lot of ground today, Sharlene. So if our listeners are intrigued and they want to learn more about you and more about the work that you do, what is the best way for them to do so?
I think the easiest way is that they can search for Santana Lab, UW, on their search engine. And usually one of the top results is our lab. Alternatively, they can go to the University of Washington Biology Department website and find my profile there, and that will also take them to our lab website, or through the Burke Museum website, where also if they look for the mammalogy collection, they also have information about our lab and links to our lab website. And all of that will also take them to my contact information if they want to contact me directly.
Excellent. Well, listeners, definitely check out these resources. Get in touch if you have any particular questions. And Sharlene, it has been such a pleasure to chat with you on the show today. Thank you so much for joining us.
It's been a pleasure talking with you too. And thank you so much for this opportunity.
Absolutely. Wonderful to speak with you again. Listeners, great to have you here as well. We'll see you next time on another episode of People Behind the Science.